1 sec. Sorry. Just a quick one. Hi, Ed Conway.
Hi, Chris Powell.
I'm just a quick announcement, then I hand it over. Okay, just to remind everybody as you're eating your pizza, that to register for CBR Research Day, which is August the 15th. All right, please do, because it's going to be a big crowd. So we really do need you to register on time, like early, because LSI is invited to this event as well as the CBR. So we need to know numbers early. All right, so please do register. Cost you nothing, and you'll get fed just as well as you do here, which is the only thing that's important, right? All right, okay, you're on. I'm going to hand it over.
Yeah, thanks. So this is the last of the virology seminars in the CBR series before summer. And it's a great pleasure to bring Chris Power here from the University of Alberta. So Chris actually is an avid skier. And at one point after leaving Johns Hopkins, where he did some of his initial training, he's a neurologist, a neurovirologist, and an internal medicine fellow. So he's one of those unique people that have combined a clinical career with a fascination and a development of science in his lab. He's got a whole lot of PNAS papers, about 200 publications. He's a fabulous person, but things were not going quite right, about 20 years ago or so. And he's actually also an incredibly, he loves cross-country skiing and madly, like marathon stuff. He wants to go and do the Baffin Island marathon ski thing. And I mean, he's tall, right? So he's got all that body build of like a Scandinavian for doing this stuff. And he was heading to trail with his wife and probably with a moving van full of his meager possessions at that stage. And this just shows how fate can swing on a dime. His wife suddenly started to go into delivery in Calgary. So they pull into the hospital and she pops the baby there, right, instead of a trail. And the neurologist up from the departmental head comes down and he heard that Chris was in labor and delivery room and convinces him not to go to trail, but to set up shop and a lab and have a position at University of Calgary. So on that chance random, event. And so for the trainees here, always, there's no luck, right? You've got to seize upon the opportunity when it comes and go for it. So luckily for Canadian science, Chris then stayed in science and was able to combine a ski career with his clinical interests and with a great lab. So when I was visiting in the early 2000s, after we made the discovery about metalloproteases cleaving and activating or changing the receptor binding specificity of different chemokines, Chris was very interested in HIV work. And that's what you're going to hear about today. A whole lot of retroviral work and HIV work. And so for those of you, the HIV virus uses the chemokine receptor CXCR4 to enter the cells. And we were working on some of those chemokines, SGF1 alpha and beta, that will bind to that receptor. And so we got teamed up there and we had a series of papers, I don't know, 6 or 7 papers, one in PNAS, and it was a really great, one in Nature Neuroscience, a really great collaboration. So being a clinician and a neurologist gives him great advantage in designing interesting animal experiments. So one of these really weird experiments that he set up, and when we're looking at HIV-associated dementia, was this mouse running around a bowl experiment. So this mouse, apparently when you put a mouse in a stainless steel bowl and run around, you just count the RPMs, right? So you make the clock and you count these RPMs of the Formula One for mice. So then Chris, being a neurologist, was able to do stereotactic injections into the brain at some point and pop into the brain, the chemokine, the cleaved chemokine and some inhibitors into the brain, just at the right spot. But when you give mice amphetamines, they start to run around the bowl. And so we get these doped up mice, plus or minus these chemokines and stuff. And so he was counting RPMs, and we're able to then validate that this chemokine was toxic and as part of the essential mechanism of HIV dementia. So if you know someone with HIV dementia, don't give them amphetamines. They might start running around, running around. So that was that was actually an interesting aside. I thought it was an interesting aside, yeah. So then he's a virologist now. He still sees patients and is on call. He started the MS Center at the University of Alberta, and he's going to talk to us today about pyroptosis and mainly on neuronal viruses. So it's a great pleasure, Chris, to have you here, and thank you for coming.
Thank you, Chris. Thanks for that very kind. Thanks for that kind invitation. I have the mic on. Yeah. Is it just turn this? Yeah. How's that? Can people hear me at the back? Great. Good. All right. Well, Chris, thank you for that very kind invitation. And I have to say, I feel honored to be at UBC. I also am awed by UBC. I'm sure everybody tells you. I went for a walk early this morning around the bog that Chris told me about, and actually it was absolutely lovely near where Chris lives. So I know there are days when it's really rainy, but this is a fantastic place to visit. So thank you, Chris, for having me. Today, a little bit about me. Chris sort of alluded, this is Lake Louise, this picture, and this is where I hang out as much as I can. Whether it's in the summer or the winter or any time of season, I ski, hike, cycle, and it's a lovely place if people have not been to Lake Louise before. And the other thing I want to do is to acknowledge that the University of Alberta is on Treaty 6 territory, and that's the traditional lands of First Nations and Métis peoples. So for today, I'm going to talk about four topics. I'm going to talk about something that we call the inflammasome-pyroptosis axis, and specifically with regard to the nervous system of the brain. And then I'm going to talk about its role in HIV infection, not in glial cells, but actually in neurons. And then I'll talk a bit about COVID and the nervous system, some of the work we've done. And finally, I'm going to talk about monkeypox infection, because monkeypox, as you all know, was a global pandemic. It's now settled down, but it remains a big issue in West Africa and Congo as well. So I'll talk to you a little about the neurotropic effects of that. Just by way of disclosure, I'm funded solely by the Canadian Institutes of Health Research, MS Canada, and also the University Hospital Foundation as well. So a quick primer in neuroscience, or just some of you, particularly if you're immunologists, you'll remember that Peter Medawar called the brain sort of an immune privileged organ. That's sort of true, except there is a lot of traffic of cells into the brain all the time. It's not isolated. The blood-brain, you may hear about the blood-brain barrier, which is this evolving concept, but it's not that protective in terms of what's going on in the blood. So there are multiple cells that are trafficking, maybe I'll do this, that are trafficking into the brain, B cells, T cells, neutrophils. And then once in the brain, they often actually, by the way, serve as vehicles for infectious agents to get into the brain. And then once inside the brain, they often will activate nearby cells, such as microglial cells, which are essentially the macrophages of the brain, astrocytes, which are the most abundant cell type in the brain, and highly heterogeneous, and of course, neurons, which are really sort of the drivers of the brain functionally, and lastly, oligodendrocytes, or myelin-producing cells. All of these cell types are permissive to infections. I'm going to talk to you a little bit about neurons and astrocytes in particular today. So what about the inflammasome? This is an area that's really been really conceptualized the inflammasome in 2002. People hear me? And then it's what the inflammasome is essentially it's a cytosolic protein complex that senses pathogens or damage and it as to cells, and it results in an inflammatory response and also regulated or programmed cell death. And that cell death is mediated through the Gasdermin proteins, which I'll come back to in a few minutes. What caught my attention about inflammasome cell death pathways is it really is a convergence of inflammation and cell death. And that's very much an issue in terms of understanding neurological diseases today. There's clearly inflammation, there's clearly cell death. How they are linked is not clear. And I'll give you an example of how they are linked today. So the inflammasome activation results in production of inflammatory cytokines, most notably IL-1 beta, IL-18, which are cleaved by caspase 1, and then upon cleavage, they're subsequently released. At the same time, caspase 1, activated caspase 1 also cleaves the prototype Gasdermin, which is Gasdermin D. And Gasdermin D translocates to the cell membrane where it forms a pore, and then upon that pore formation, there's extravasation of cell contents, as illustrated here. This is from a review of one of the students in my group, Bree McKenzie. And those extravasated those extravasated molecules are sort of a forward-feeding process to drive inflammation in the brain, along with the cytokines that I mentioned earlier. So that's sort of the background to those of you who aren't familiar with inflammasome biology. It's clear there's a link, and that's what prompted us to sort of link inflammasome biology to pyroptosis. Pyroptosis is quite distinct, though, from other types of cell death. It's a programmed cell death, but it's different from necroptosis, which is dependent on a different set of proteins. And then, obviously, with time in apoptosis, cells may actually die and there may be extravasation of cell contents, but it's quite distinct. There are 12 types of programmed cell death today, or even more. There's a movement in the field to try to sort of, that they are all linked, and that's true to some extent. People have coined the term panoptosis, but I still think that there's a role, and I'll show you examples today, of where individual molecules actually drive different types of pyroptosis, or drive pyroptosis, not necessarily different types. So that's a bit of background. So that brings me to the first virus I'm going to talk to you about today, which is HIV. And as Chris alluded to, that's a virus that is, it's a retrovirus. Retroviruses really like to infect the nervous system. And for anybody who's a young person interested in viruses and science, there's a whole range. Not only does HIV remain a contemporary problem, which I'll talk to you about in a moment, but there's the monkey virus, which I'll talk about today, our non-human primate virus, SIV. And then there are other viruses. For those of you who might be horsey, there's a virus in horses. It's an issue in wild horses. Those of you who are cat owners, there's a virus called FIV, which is actually also in local cougars here in British Columbia and Alberta. But you may know that about 8% of the human genome is comprised of retrovirus sequences. And the extent to which that they contribute to disease is unclear. My group's been interested in the role of endogenous retroviruses in multiple sclerosis, which I won't talk to you about today, but I think it's a pretty hot area. And so if you're looking for new areas to work in, there's a lot of interest in endogenous retroviruses. These such as HERV-W, HERV-K, and actually even MMTV, which is a mouse virus that also appears to infect humans. So that's a bit of background about HIV. And yeah, that's the other thing I wanted to, there is something distinct about the lentiviruses, i.e. HIV and FIV and SIV. They like to infect macrophages. And that's different from some of the other viruses. The other human retrovirus is HTLV1 down here at the bottom, which prefers to infect T cells exclusively. Maybe a little bit of astrocytes, and it interestingly causes brain disease as well. Okay, a bit about HIV. So remember the pandemic started in the 1980s. It was and then obviously spread globally. But the understanding of HIV has evolved also concurrently. And at one time, we knew, yeah, sure, there was, when people became infected, there was a tremendous viral burst, and then it resolved, and then there was a drop in CD4, and with time, that the CD4 cells dropped. But that's changed with the advent of antiretroviral therapy. So just like viruses, like vaccines were really a medical miracle for COVID, antiretroviral therapy was also a medical miracle. I mean, we went from people, frankly, dying, not knowing who was going to come to clinic, and with the advent of antiretroviral therapy in 1996, it was a real game changer. And so what happened was antiretroviral therapy is now, instead of being given it when people are really sick, at this stage, we now introduce antiretroviral therapy early on, right at the onset of infection. And that's changed the whole face of the disease. So the life expectancy for someone with HIV today is pretty much the same as the general population, assuming they start antiretroviral therapy at an early stage. And that's really been remarkable and fantastic for people who are infected or affected by HIV. Nonetheless, there are over 38 million people infected with HIV. About half of those people receive antiretroviral therapy, which is a pretty sobering thought when you think about it, because untreated HIV is a lethal disease. That's indisputable. So if you're not getting treated, you're in trouble. In Canada, there are about 71 million people with HIV, or 71,000 people with HIV infection. And remarkably, and this is what keeps me busy today, people still have neurologic disease. About 25% do. And that's validated not just from our own cohorts, but also big cohorts in the US. There's a very well-known cohort called the MAX study, where they report that it's actually higher than 25%. I find that a little hard to believe, but I think we have better access to antiretroviral therapy in Canada than in the US. Okay, so there's another thing you need to know. We can essentially eliminate virus from the brain, from the blood, very effectively, so that people have undetectable virus in the blood by very sensitive tools, RT-PCR. But we were concerned because we still see brain disease. So we decided, this was a graduate student, or is a graduate student in my lab, Naz Mohammadzadeh, where Naz said, you know, I'm concerned, you see neurologic disease, is there still virus in the brain? And to make a long story short, what Naz showed that even amongst people who were on antiretroviral therapy for years, I mean years, like upwards of 10 years, she still found that in fact, people had viral RNA, DNA, and indeed integrated DNA in the brains of people who were treated with antiretroviral therapy right to the time of death. The immune response wasn't that helpful, but it was very clear that we could see viral genome, and in fact, we could also see viral antigen in the brain. This was subsequently, well, Naz did it in human autopsy material, and then we turned around and in collaboration with Jérôme Estiquoyer at Laval, we then actually did it again in non-human primates infected with SIV. We treated animals with a conventional antiretroviral therapy and basically saw that the viral loads were the same in brain, presence or absence of antiretroviral therapy. Again, a disturbing finding, but nonetheless, that does explain why we continue, or may partially explain why we continue to see brain disease in people with HIV. The other thing is, importantly, this has been validated. This is not a one-off. Melissa Churchill at Monash in Australia, Donna Gabuzda at Harvard, both have published very similar data, and it's also been validated in the another SIV group, University of Pittsburgh, showed the same thing. So this is a real issue that's not going to go away. So people who are interested in virology, this is a new reservoir, or not a new reservoir, but it's a reservoir we really need to target in terms of therapies. Okay, so what about brain disease? Chris mentioned HIV-associated dementia. Thankfully, we don't see it with the same severity that we did 20 years ago when Chris and I were collaborating. But nonetheless, we see a syndrome called HIV-associated neurocognitive disorder, where people have difficulty with memory, decision-making, some behavioral changes, tremor, Parkinsonianism. That's usually in the context of prior immunodeficiency. And as I told you, there's virus in the brain. And the result is damage to neurons. Here's someone with AIDS who did not have hand. and someone with hand who had frank dementia. And you can see here that the neurons look dysmorphic and there are fewer of them. And it's not just big cortical neurons, but it's also subcortical GABAergic neurons. We think the data is pretty strong that the first step is synaptic injury and retraction, and that eventually results in frank cell death, which drives the clinical phenotype. That's also all in the background of chronic inflammation. It's very likely that this inflammation drives or contributes to the neuronal injury, directly or indirectly. We know about, I told you a moment ago about inflammasomes. We've known that IL-1 beta and IL-18 are players in the pathogenesis of hand. This is work from a former postdoc, John Walsh, who showed that in macrophages, there was lots of IL-1 beta production, and we saw other IL-18 was also induced. And then more recently, Will Branton, who's actually our senior technician, reported that in fact we saw multiple inflammasome protein or genes that were induced in patients with hand. Caspase-1, IL-1 beta, IL-18, and indeed even gasdermin D. So that was interesting, and that's really just fuel to drive us to pursue inflammasome biology further. But I need to tell you about a viral protein that we've worked on for many years in collaboration with Eric Cohen at the University of Montreal, the viral protein R. It's a really interesting protein. It induces cell cycle arrest. It's required for macrophage infection. So that's pivotal for infecting the brain because the virus, HIV, chiefly infects microglial cells. So VPR is important for that. It's secreted, and it's also neurotoxic. We showed that multiple times in vitro. Then we made a VPR transgenic mouse many years ago that was driven by a macrophage gene, and then we found in those transgenic animals there was evidence of neuronal loss, and also there was evidence of apoptosis through caspase-3 detection in those neurons. I was left a little concerned about this. We published the data in JNeuroSci, but I wasn't that convinced that cell death looked like, although we could see caspase-3 in the neurons, I was not convinced it was really apoptosis. The cell morphology didn't look what I was expecting, like other neurotropic viruses. So this was in neurons, so it was not a direct infection. That was years ago, a long time ago. And then along came our interest in gasdermins. So let me just tell you a little bit about gasdermins. They're an interesting group of proteins. We don't really know what they do from an evolutionary perspective, other than this cleavage function, which certainly serves to get rid of infectious agents. But I don't think it's their sole function. I can talk about that afterwards. But there are multiple gasdermins. Gasdermin C, not really expressed to any extent in the brain. Gasdermin E, which I'll tell you about more in a few minutes. Gasdermin B, which I'll also tell you about a little later with another virus. And then also gasdermin D, which is the prototype gasdermin, which is expressed in macrophages and really been the best studied of all of the gasdermins. Gasdermin E, though, is different. And I want to get your attention. Remember, I just talked to you about caspase-3 expression in neurons in our transgenic mouse model, but caspase-3 actually is responsible for cleaving gasdermin E. So it's a bit different from the other gasdermins where it's caspase-1 that's responsible. So I told you that neurons damaged in HIV and that subserves the clinical phenotype of HIV dementia or hand. So we didn't know which gasdermins were expressed in which neural cells, except for gasdermin D in microglia. So we looked to see we were fortunate to have primary human neurons and microglia and astrocytes. These are actually an oligodendrocyte cell line. And we looked to see which of the gasdermins were expressed in which cell types. And indeed, gasdermin E, which I'm going to tell you about in a moment, a bit more, was most principally expressed in neurons, as illustrated here. And gasdermin B was principally expressed in astrocytes, which I'll come back to that in a few minutes. So knowing that gasdermin E is expressed in neurons, knowing that neurons undergo cell damage in the context of hand, were the two indeed linked? was a question that we started on. This is the work of another graduate student in the lab, Jason Fernandez, who is a fantastic scientist, just as the Naz and Bree were. And first thing Jason did is he said, okay, can I find Gasdermin E in the brains of people with hand? Indeed, he did. As illustrated here, you can see that this is from people without HIV, people with HIV no hand, and people with hand who tragically died. And you can see that there's Gasdermin E in neurons. They're co-localized with a neuronal marker. And in those same neurons, we see caspase 3 illustrated here. We then went on and looked by Western blotting, and I won't spend too much time because I know Western blots don't keep people awake. But nonetheless, it's important you know that we did see evidence of neuronal injury as evidenced by beta-3 tubulin suppression in patients with hand. This is from the frontal cortex. And accompanying that, neurodegeneration, there are loss of this neuronal protein. We actually saw more cleaved Gasdermin E as illustrated here on the Western blot compared to patients with HIV infection without hand or controls. And Jason has graphed that over here, showing that indeed there's less beta tubulin in the hand patients, and there's much more of the cleaved Gasdermin E. So we wanted to explore this further in vitro. So Jason, we were very fortunate to have human neurons that we can work with. And so he wanted to say, could we reproduce this phenomenon in vitro? Indeed, when we exposed human neurons to VPR over time, we actually saw an induction of Gasdermin E as illustrated. Certainly here, the cleaved form is here, but it's induced here. And when he looked concurrently to see if there was LDH release, indicative of cell lysis or pyroptosis, he also saw a marked increase of LDH. And that was also accompanied by another marker of cell lysis called cytox, or green nucleic acid stain. So that validating the LDH assay. So what do the cells look like? Well, I think you can see here with cells that are expressed or that are exposed to VPR. Remember, I told you VPR is secreted. Indeed, compared to the unexposed or mock exposed cells, the neurons look really unhappy. They're shrunken. And indeed, Gasdermin E, you can see here, compared to the mock exposed, the Gasdermin E accumulates at the cell membrane, and overall they look unhappy. And of course, as I said a moment ago, they actually go on to die. So what we wanted to know, can we modulate this process? I'm a clinician. I'm always interested in trying to treat diseases. And so we looked to see whether or not this caspase 1 inhibitor, because we had a suspicion caspase 1 was also part of the process. We know that it's also induced in neurons, just like caspase 3. So we want to know, if we block caspase 1, would that influence what we're seeing? And indeed, when we exposed cells to VPR and with VX765, sorry, the text is small there, the VX765 actually reduces the cleavage of Gasdermin E. It also, as a result, reduces the cleavage of caspase 3 downstream, and suggesting that caspase 1 is activation linked to caspase 3. And that's illustrated here graphically. Then we went on and actually, just to be sure that this was real about the caspase 3, we looked for PARP cleavage, and indeed you can see that VPR-induced cleavage of PARP, and then that was blocked partially, not completely, partially by VX765, suggesting that the two caspases are indeed linked. It's always nice to see something visually. So Jason actually developed an assay where we can actually look at neuronal processes. These are, again, primary human neural cells or neurons. Controls, cells exposed to the caspase 1 inhibitor. When they're exposed to VPR, they look terrible. There's obviously marked loss of cell bodies, retraction of processes, and there's a partial retraction when we, or partial recovery or protection when we expose the cells to VX765 in advance. And that's illustrated here. We obviously reduce LDH production, and also the number of cellular processes was restored with VX765. It's not full by any means, but it's certainly better than no treatment at all. So again, sort of underscoring the relationship between caspase 1 and caspase 3. So I'm just going to move on in the interest of time. So we also went on and validated this in a monkey model. It's great to use autopsy tissue and do in vitro studies, but it's really imperative that you try and validate it in a good model. And in the world of HIV research, the SIV model is amongst the very best. There are other models like humanized mice, but I would say this is perhaps the Cadillac model for sure. And not surprisingly, when we looked in SIV infected animals, we found SIV genome in the brains. We also both SIV Pol as well as SIV VPR. And then those animals with neurologic disease, they had behavioral features abnormal behavioral features that were infected. Those animals, at least 2 of the 3 of them, had marked reduction of beta tubulin. And when we looked for Gasdermin E expression, it certainly was induced at the full length, and also the cleaved version of it was also apparent as illustrated here. And that's graphed, the full length is graphed here. I have to say, full disclosure, this looks like it's the cleaved product, but it's a different species, and we don't have an antibody that I can be 100% certain of. That really probably requires more work in the area of probably mass spec. We also then went on to look to see, can we see the same story that we saw in humans and in vitro, and indeed in the animals with SIV infection and brain disease, we could certainly find Gasdermin E in neurons, which co-localize with caspase-1 as illustrated here. And these animals also had lots of virus in the brain as well. So let me just summarize this first section. Gasdermin E is expressed in neurons. It undergoes proteolytic cleavage that appears to cause neuronal pyroptosis in models of hand. And caspase-1 seems to activate the caspase-3 Gasdermin E axis, resulting in neuronal pyroptosis, and blocking caspase-1 seems to have a therapeutic effect. And that's just a schemata of what we think is happening. I didn't tell you this part. VPR, we published previously that VPR activates one of the major inflammasome proteins called NLRP3. That was from a postdoc several years ago, published that work. Okay, so that's enough about HIV. I'm going to switch gears. I'm going to talk about another important virus, and that's COVID. So I think many people know this already, that COVID is associated with neurologic disease. We wrote a review on this with Aaron Balcom, who's now one of our senior neurology residents. And basically, there's a range of neurologic phenotypes that you see in the context of COVID-19. Many of you know there's a loss of smell, but patients get encephalopathic, stroke, seizures, meningitis, and even the peripheral nervous system is affected. And there are also some post-viral syndromes as well. There is inflammation in the brain. Early on in the pandemic, a large German consortium published that when you looked in the brains of people who had died of COVID-19, indeed, there was lots of microglial activation, as illustrated here, and it was throughout the brain. There was a lot of controversy about the presence or absence of virus in the brain. And I'm going to come back to that, because I think that underscores an important point. The pathogenesis, though, we were seeing, and we saw weird syndromes too, like weird movement disorders, and that I'd seen in children, but I'd never seen in adults. So it was really an eye-opener in terms of what was happening, not just in terms of people having lung disease, but also brain disease. So the mechanisms of how COVID injures the brain is really unclear. We know, I'll come back to the neural invasion in a moment because it's controversial. There's a lot of interest in autoimmunity. Indeed, there's a lot of evidence for it. I just showed you there's inflammation in the brain in terms of microglial and astrocyte activation. Is it all driven by systemic inflammation? It's probably a factor. There's also a lot of interest in endothelial cell damage and disruption of the blood-brain barrier. One group of, or one area that our group at the University of Alberta is really interested in is peroxisome biology, and that's really by virtue of history. Rick Ratchabinski was really sort of a world leader in peroxisome biology, and so my colleague Tom Hobman wanted to apply Rick's sort of expertise in this area, and he started to look to see whether or not peroxisomes might be injured by different viruses. And Tom has a legion of papers on different viruses that affect peroxisomes. And I'll tell you why this is relevant to SARS-CoV-2 in a moment. But indeed, Rick and Tom actually published a paper showing that when they infected neural cells, they did see peroxisome injury. And that was an in vitro paper. So we were interested in chasing this in vivo using different models that were available to us. But why are we so interested in peroxisomes? Well, they're kind of this first cousin of mitochondria, but they're really interesting organelles because they express lots of antiviral proteins. They express MAVs, they express IRF3, IRF1. So they have a very strong impact on regulating virus, and also they are actively targeted by viruses. Tom showed that HIV VPU damages all peroxisomes. Other groups have shown that other flavivirus proteins can damage peroxisomes. So that made sense for us to think about SARS-CoV-2 and whether or not it damaged peroxisomes in the brain. Remember, the brain is also loaded with peroxisomes. There are neurons, oligodendrocytes. So it made sense for us to look. So we started out with two questions. And when we got started, people were saying that there was no virus in the brain. That's not really the case. And so we wondered, despite minimal virus in the brain, does SARS-CoV-2 actually induce neuroinflammation? And I'll make a long story short, yeah, it sure does. And secondly, was COVID-19 associated with suppression of individual peroxisome proteins? And I'll tell you about that in a moment. The first thing, though, is the issue of infection of the brain. So there was a lot of controversy. Many and friends were and colleagues were writing papers saying, no, we can't find any virus in the brain. And so we were fortunate. We had an assay running for using digital droplet PCR. We looked for lots of viruses doing that. And indeed, what we found was that, sure, we can find SARS-CoV-2 in positive samples and lung tissues and infected cells. But you know what? We also found it in a subset of brain tissue from patients who died from COVID-19, as illustrated here. So we found it in the cerebrum, we found it in the olfactory bulb, and we also found it a lot in the brainstem. And that's an interesting issue in itself because the brainstem affects many neurotransmitter systems. Indeed, when we really looked in a couple of patients, we actually found viral antigen in the brain. It was co-localized within neurons, and we found multiple, as well as astrocytes. We found multiple viral antigens were expressed, as illustrated here. And we went on and verified that in vitro, showing that indeed SARS-CoV-2 replicated in astrocytes and, well, obviously in vero cells as well, to a greater extent in vero cells. So we found that indeed there was a rationale for finding virus in the brain in that it was tropic for astrocytes. Around the same time, just as our paper was coming out, there was a group from the NIH, Stein et al. Published a paper in Nature, and they found virus in the brain upwards of 9 months after people had acute COVID-19, underscoring the fact that indeed there was really virus in the brain. So what about our other questions about inflammation? Yep, there is lots of inflammation in the brain. We saw inflammatory cells in the brain. These are individual cell markers in patients these are our negative controls, but in our COVID patients, we found activated T cell or presence of T cells. We saw complement upregulated, indicative of activated astrocytes. We also saw CD18, which is on macrophages, and MHC class 2, all induced in the brains of patients with COVID. And we saw lots of the inflammasome markers were also upregulated in the brains of people with COVID-19. That was at the RNA level. Oh, yeah, and we saw individual cytokines and chemokines were upregulated in the brains of people with COVID-19 in the cerebrum, olfactory bulb, and the brainstem. That was good. That was at the RNA level, but I wanted to be sure, it's great to look at RNA, but we wanted to be sure we could see it at the protein. So in collaboration with Nathalie Arbour at the University of Montreal, we actually looked for cytokines in the brains using Luminex. And indeed, we found that upregulation of IL-18 in the white matter of patients with SARS-CoV-2 infection or COVID-19. And we actually, interestingly, saw a suppression of IL-19 in the cortex. This is in white matter, and this is in the cortex. Bit counterintuitive, but nonetheless, we did see upregulation of IL-18. So what about the peroxisome story? So we actually went on and in fact, did look for different sort of established peroxisome genes. This is at the RNA level. We saw some upregulation of some of the individual peroxisome genes, associated genes. That was illustrated here. And then there was suppression of others, depending where we looked. Olfactory bulb, they were increased. Brainstem, it was reduced. But then to validate that, we went on and did Western blotting. And this is really sort of a prototype protein, PMP70, is suppressed in the brains of patients with COVID-19. And other proteins such as PEX11B was also suppressed in the brains of patients with, not non-significantly, but there was a trend. PEX14 was also down, suggesting that, yep, there's damage to the peroxisome proteins in patients with COVID-19. We then went on and looked at hamsters and essentially found the same story. We looked at different time points, day 4, day 7, day 14, post-infection. And in fact, we found by day, it was interesting when there was a delayed effect. By day 14, we saw a dramatic increase in the cytokines in the brains of animals at day 14. We also found virus in the brain, albeit not very much, actually less than we found in humans. And we did it, we also looked for different peroxisome proteins, and PEX3 and also catalase were suppressed in the brains of the hamster model. I'd have to say that the hamster model is not great, and we now have a mouse model that we use that's much better. The other thing I want to just mention, I have an eclectic team, and I just want to acknowledge that this is actually an example for you young people. This was a guy, Andrei Rakowski, who was a technician in the lab, really great guy, and now and he got turned on to science, and now he's in third-year medical school, and I'm hoping he's going to be a neurologist. He's a great guy, and I continue to collaborate with him. So, this was like an about face for him in life. He'd sort of kind of was giving up on research, and then he got this project, and bang, he was turned on and went to medical school, and he's been writing papers all the way through medical school. So I don't get discouraged. He's really a strong. And actually, sorry, this is published already. It's in the Annals of Neurology. So in summary, I can tell you that SARS-CoV-2 infects neurons and astrocytes in a subset of patients, particularly in the brainstem. And that's interesting because the brainstem controls a lot of neurotransmitter pathways, such as cholinergic pathways. And when I see patients in the long COVID clinic, these patients are really suffering with depression, anxiety. They have cognitive symptoms as well, but the mood issues are overwhelming. And I think that may be linked to the brainstem infection. The other thing is COVID-19 is associated with a very robust immune response in the brain with limited detection or replication. And finally, peroxisome genes and proteins are all suppressed. And so we're now kind of chasing this as a therapeutic strategy to see if we can improve the outcomes for patients with COVID-19. About long COVID, I'm not going to spend too much time. I'm just going to say a little bit about it. It's a huge problem. There are about 1.4 Canadians who have it, and it's really ill-defined. It's not clear. There's like 200 symptoms associated with it, and the disease mechanisms are even more obscure. But it's a major issue. As I told you, mood issues are a big problem. Fatigue, chronic pain, and so and it remains a big issue. And should we have another pandemic, it's going to be a real problem because this is very highly disabling for patients. The other thing that's also a sobering point is this next slide. This is work we just published a few weeks ago. This is from Candice Marstairs, who's a neurology resident at the University of Alberta, where she actually looked, this is from a cohort of about 350,000 patients that we have in Alberta. This was what we call health services data, and we looked epidemiologically at the risk of developing encephalopathy, dementia, spinal cord disease or myelitis, or seizures or epilepsy after COVID-19. So we had exact time of when people had a positive or negative PCR test. Each group was a case control analysis of about 180,000 in each group. And we looked, and indeed, the risk of delirium or encephalopathy was higher in the patients with COVID. The risk of dementia, which is really disturbing, but not without some precedent. Other groups have suggested this, also higher in patients with COVID-19. Spinal cord disease was also increased, and likewise the risk of seizures. So this is a concern, and we've, I won't have time today, but we've been developing an animal model to look at the underlying mechanisms. We do also have a cohort, and not surprisingly, and I'll just finish the COVID part here, but not surprisingly, those patients with long COVID have marked induction many months, even years after they've had acute COVID, of inflammatory markers in blood. These are serum biomarkers. Not surprisingly, they're inflammasome-related, IL-1 beta, IL-18. They're all induced in blood of patients with long COVID compared to patients who are controls. The controls are people who had COVID but did not develop long COVID. And it's worse when we break it down further in terms of brain disease. People have neurocognitive disorder related to COVID. That group has particularly high inflammatory markers. But it's a range of markers, not just inflammasome. TNF-alpha is increased, IL-6, and also the neuronal protein tau is also increased, indicative of neuronal injury. So I'll leave the COVID part there, but I think we're going to hear more about this going forward. There's a federal report on long COVID, and it remains a major challenge. Okay, in the last few minutes, I'm just going to talk about an emerging infection. So we live in an era of emerging infections. I don't need to tell people in this audience. I started documenting this in '81, but they obviously preceded. There have been many infections in that time. The most recent one is, well, actually, this is a bit outdated because H5N1 is also neurotropic. But monkeypox is a big issue. And it was kind of scary for a while because it was really, it spread across Europe and North America very, very quickly. But it was and it remains an issue in Central and West Africa. And perhaps most disturbingly, it's a very big issue for children. What is monkeypox? It's an orthopox virus. It's related to smallpox, and it's also related to vaccinia. And actually, it's quite. I'm going to show you where we've compared monkeypox to vaccinia, and the differences are quite striking. Monkeypox is also associated with neurologic disease. And this is actually a paper. This is actually, there have been multiple papers now suggesting that people get seizures or epilepsy, encephalitis, or just frank delirium with monkeypox infection. And it's suggestive of infection of the brain. It's because it's actually fairly focal, as you can illustrate here, suggesting maybe the virus gets directly into the brain. So this is work from a postdoc in the lab, Hajar Miranzadeh. And Hajar asked two questions. She said, does monkeypox, and oh, by the way, one point of clarification, mpox is the disease caused by monkeypox, and sometimes there's a bit of uncertainty around that. But, so I'll just talk about monkeypox virus, because I'm just going to show you in vitro experiments. So Hajar asked the question, does monkeypox indeed infect neural cells? In fact, well, I'll tell you about that after we have time. But yeah, so that was her first question. Her second question is, what was the inflammatory response? And what were and were there any specific cell death mechanisms activated? So the first question is, okay, does monkeypox infect neural cells? And for this, we had to collaborate with Ryan Noyce and David Evans, who are both pox virologists, because this had to be done in the BSL-3 facility, just like the SARS-CoV-2 work. And so we really needed good collaborators, and Dave and Ryan are sort of prototype pox virologists. So very fortunate to have them. And what we found right off the bat is that, in fact, monkeypox really prefers to replicate in astrocytes, as illustrated. It doesn't really, it infects microglia a little bit, does not infect neurons at all. And maybe in oligodendrocytes, this oligodendrocyte cell line was a little bit permissive, but it was very clear. This is all done by plaque assay. It was very clear that monkeypox infected astrocytes. So did vaccinia, as illustrated here. This is a specific strain of vaccinia. It also liked to infect astrocytes as well. And you can actually see viral proteins expressed in the infected cells for both viruses. But we knew right away when Hajar did these experiments, she said, you know what, the cells are dying, like they're dying fast. And pox virus is a little different from SARS-CoV-2 or HIV. They really overwhelm the cell. They just sort of take over the cell so right away we knew we were onto something related to cell death. We also were fortunate to collaborate with Olivier Julien, who's a proteomics expert at our institution, and we were looking for immune responses. And truthful, we found a lot induction and suppression of multiple proteins in astrocytes that were infected by monkeypox. Some of them were interesting, but did not really give us a clue to what was going on. But we did see some interesting protein. This is a protein called autotaxin, which is induced by monkeypox. We see it in injured cells and other diseases. S100, many of you know that's expressed in astrocytes, and it was highly induced. Just indicative of inflammation, but not really a clue to the specific mechanism. So we went on. I told you that Hajar told me the cells were dying. So we went and looked for evidence of cell death. And indeed, the cytox, or the green nucleic acid assay, showed that it was markedly induced in monkeypox-infected cells compared to vaccinia cells. These is in astrocytes. And likewise, LDH levels were sky high in the monkeypox-infected cells compared to vaccinia. There was a little bit of cell death with vaccinia, but much less. So then we went on to say, okay, knowing that we see cell lysis where the caspase is induced. And this is actually a Western blot of monkeypox-infected cells and vaccinia-infected cells. And the full-length pro-caspase-1 was markedly suppressed. And I don't have the lower blot. It didn't show up that way. It's not that pretty. But we saw some induction of caspase-1 in these cells. The problem is the cells are dying so quickly, I think. So we don't see the lower fragments of caspase-1 in these cells. Nonetheless, caspase-1 was highly induced. So this brings me back to the Gasdermins. Knowing that caspase-1 was induced, and knowing that Gasdermin B was principally expressed in astrocytes, we started to look for Gasdermin B in infected monkeypox-infected astrocytes. And Gasdermin B is kind of interesting. It's specific to human, or to rodents, or to primates rather. It's not expressed in rodents, so we couldn't use a rodent model of monkeypox. It's a substrate for both caspase-1 and caspase-4. And this is actually from Judy Lieberman's. This is just showing how caspase or Gasdermin B is actually cleaved by caspase-1 and 4. And it generates this N-terminus, which goes to the cell membrane, just like Gasdermin D, I told you about earlier, and results in pyroptosis. So when we looked for the presence of Gasdermin B, indeed we saw it in infected cells. And then we saw the cleaved form as illustrated here and we saw both the N-terminus, but we also saw the C-terminus as shown here in the monkeypox infected cells, but really not to the same extent as we saw with vaccinia illustrated here. So we see both the N and the C termini of Gasdermin B. So we'd like to block that if we could, right? Like this is sort of an instinct that, as a clinician, I want to try and do something about this clinically. So we knew from Kate Fitzgerald's work, she had a paper in Science showing that Gasdermin D is actually blocked by dimethyl fumarate. Dimethyl fumarate is a drug that is used for treatment of psoriasis and actually also multiple sclerosis. So so we have a lot of familiarity with this drug. So, and what it does is it actually blocks the cleavage of Gasdermin D by caspase-1 through interrupting succination. So we thought, okay, we know that caspase-1 is involved in cleavage of Gasdermin B. Is it possible that dimethyl fumarate actually also blocks Gasdermin B activation? And indeed, when we actually pre-treated cells with DMF and then infected them with monkeypox, you can see that the C-terminus of gasdermin B was markedly reduced. This is a better antibody, quite frankly, and that's why we used it. But dimethyl fumarate knocked it down, and actually this is a dose-dependent effect. I'm not showing you here, but we saw a dose-dependent effect. And the monomer methyl fumarate, monomethyl fumarate, really didn't have the same effect as dimethyl fumarate, and that's illustrated here. So what do we think is happening? We think that monkeypox infects human astrocytes and causes changes in the morphology and eventual cell death. It results in activation of gasdermin B by caspase-1, which we can block by dimethyl fumarate treatment. And thus, we think that pyroptosis may underpin the development of monkeypox neurovirulence. And that's just illustrated here in this schematic. And that's Hajar Mirzandeh who did all of the work. So I want to wind it up. So none of this work could be done without an absolutely wonderful group in my lab, some of whom I've highlighted today: Jason, Naz. I didn't talk about Niall's work in multiple sclerosis, Will, and Andre, and others in the lab, and including clinicians as well as trainees, and then a really fun and great group of collaborators. I mentioned Tom, Jérôme, Eric, and then a group in the Netherlands and also the US. And of course, the funding agencies without whom we could not do any of this research. I'm very grateful to. So this is just another part of the Canadian Rockies. And if you guys are interested, that's how to find me. Now, I want to be straight about me. I'm from Alberta, right? I'm from Edmonton. Who's going to win tonight? And guess where we're going? I'll start. No, and I'm willing to challenge anybody. That was a long time ago. Thank you for your attention.
Thank you, Chris. How does viral protein induce? What's the target?
Yeah, so it forms a pore. It forms a pore in the cell membrane, and that pore is permissive to potassium influx, which activates NLRP3. That's what we think. And other people have shown that. Have shown not so much the activation of NLRP3, but they've shown that VPR forms a pore in cell membranes.
I thought you said also it also then, I guess, that subsequent thing gasdermin.
Yeah, it does in vitro. So it activates that cascade. I could have spent more time. It activates the assembly of NLRP3, which serves as a platform for activation of caspase-1. I don't need to tell you this, you're a protein scientist, but and then as a result, caspase-3 or caspase-1 or 3 can then cleave the gasdermins. So yeah, and we've shown, and other people have shown, if you change this sequence of VPR, you can change the biology. And in fact, it's even associated, the sequence of VPR is associated with the clinical phenotype as well.
The pores that formed initially, that's more this, but not as an ion channel.
Yeah, and it's permissive to potassium. Yeah. One other thing I didn't talk about, for those of you interested in inflammasome biology, typically it requires a signal 1 and a signal 2 to activate the inflammasome. But because VPR actually activates signal 1, i.e. the pore formation and activation of NLRP3, that's enough to initiate the cascade. Yeah. Other questions? Yes.
So I'm wondering, in these monkeypox infections, do you see caspase-1 activating caspase-3 and then that cleaving gasdermin B?
Yeah, it's a good point. We do not see activation of caspase-3. I didn't talk about that, but we don't see activation of caspase-3 in the monkeypox, unlike what we saw with HIV. So gasdermin B is not dependent on caspase-3. It's only dependent on caspase-1 for cleavage.
Okay.
Does that make sense?
Well, typically it's caspase-3 that's known to activate it by cleavage and not caspase-1, right? So that's what I'm wondering about.
I'm glad you raised that. Our collaborator, Neil Alto, was also really concerned about it. And of course, the other protein that cleaves gasdermin B is granzyme A, right? Yeah. But we could not show that, we didn't see any evidence of caspase-3 activation in astrocytes. I'm glad you highlighted that. So and that was a bit of an issue, but we looked really hard for caspase-3 activation. We also, by the way, looked for granzyme A as well, and we could not find it in astrocytes. And that's pretty well known, I think. That's
Yeah.
pretty widely accepted.
So something else must be
Yeah.
cleaving it, and it's
Yeah.
just unclear yet.
Yeah.
Okay, cool. Thank you.
Yeah, well, we think it's caspase 1.
Yeah.
Okay,
Have you tried it in like with recombinant protein or
good
like
point.
to
We've
see
not
if
done
it
that.
if it
We've not done
cuts?
that. That's a
No.
good point. Yeah.
Okay. Okay.
Yeah, I don't
Thanks.
think we've done that. Yeah. Yes, Selena. Hi.
I was just curious about the HIV in the brain. Are there any differences with the level of the virus
Yeah. So, yeah, that's how I got started, like in the dark ages, is looking at sequencing virus from the brain. Yeah, and we've shown, and many other groups have shown, that there's compartmentalization of the virus in the brain, typically macrophage tropic CCR5 dependent virus, but more people have even gone further and said that there are individual sequence differences, particularly in the V3 loop. We've shown it in the VPR, and I think people have also shown it in NEF as well. Yeah.
Can you scare us being a center for blood research, like I'm already
Yeah.
viruses should we be concerned about and how to what things aren't we doing?
What's on the horizon? Where's Francois? Francois educated me a lot this morning, although I think you will have heard in the news too. I think H5N1 is probably is the most worrisome. There is a virus that, yeah, I think that'll be an issue because they're finding it in milk, right? And cow's milk, right? There is another virus that I won't have time to talk to you about, but if anybody's interested, Selena knows about this, is Peggy virus, which is another flavivirus that is ubiquitous, that can cause encephalitis in immunosuppressed patients. But I won't have time to talk to you about that. It's something, it's another project in the lab. It's in the blood supply. Yeah, I don't know. I wouldn't, actually, I can't. I'm not sure, Chris. I mean, that would be a great proteomics question to ask, right? Wouldn't that be a good thing to do, I think? Because, I mean, you want to know if there's, I mean, sure, it's great to have viral genome, but if you can find proteins there, that would be even better. Yeah. Good. Yes, Francois.
General question, clarification. What is it you know about? Is it like cargo, vesicle? So what's the state of the circuit?
Good question. So it is secreted. It's released. We can detect it in blood, free VPR, and also in spinal fluid. I mean, you know, most of these accessory proteins have some kind of function to suppress host responses, right? And VPR, of course, it's known for sort of shepherding the pre-integration complex. But it also, I guess, there are probably other functions, other sort of host restriction factors that it interferes with as well. But I'm not, other than that, I can't really speak to it. Do you have a thought?
Just curious, for example, for flaviviruses, and each one found positive and positive could be longer if there may be a similar phenomenon.
Yeah. That's a good point. I don't know. I can't speak. That's a very good point, actually. I'm not sure. Good thought. Yeah.
In the brain section we're doing from the code
Yeah. Yeah. The neuropathologists did report on some of the cases actually having sort of, you know, brain hemorrhage as well. So hemorrhage as well as clots. Yeah. So for sure. So, but you know, well, as I alluded, yeah. So I think, I mean, we know that there's endothelial cells are injured, so there's going to be cells are going to sludge, particularly in small caliber vessels. So, but precise mechanism, I'm not sure. Yeah. I would suspect what's happening is that cytokine storm that happens inflames endothelial cells and nearby cells like pericytes, and as a result, anything flowing through a narrow caliber vessel will sludge and stick to it. So you get clots and sometimes hemorrhages as a result. Yeah.
Do I stay pressed? Okay, you guys can hear me okay? Okay, awesome. I just had a question about COVID. From a last class, I kind of knew that it's somewhat related to ischemia, like a lack of blood to the brain. But now that you're mentioning hemorrhages, is there somewhat of a relation to that when you're mentioning that there's like endothelial damage? Is that related to both? Like, could hemorrhages and ischemia happen?
Yeah, for sure. Yeah, absolutely. So and I'm not a stroke scientist, but for sure when leukocytes and platelets clump or within blood vessels, you get ischemia, right? It interrupts blood flow. But on top of that, you can also, if it's bad enough, you can also damage the blood vessel, and as a result, there's hemorrhage into the brain. Does that make sense?
Yeah.
So so we can see hemorrhages on MR of patients. We also see ischemic lesions as well.
Okay.
So.
As a neurologist, why would some patients need to expel like that?
Yeah. Yeah. No, it's peripheral. The virus infects what are called the sustentacular cells, and those cells don't regenerate very readily. So that's why people have persistent anosmia. And then the other thing is the nose is a great way into the brain. So all our hamsters and also our mouse model, which I didn't talk about, we infect intranasally. And so I think that facilitates it getting into the brain a bit. We've done other viruses will get to the brain that way as well.
Hi. So VPR from HIV is obviously involved in this process. What about VPRs from endogenous retroviruses?
To my knowledge, I don't think there's an equivalent. It's the structure of lentivirus is pretty different. The endogenous retroviruses are incomplete, right? They're not replication competent. And I don't know that they express any similar accessory proteins. They obviously have a pol, an end, and a gag, and then some, but I don't think they function the way lentivirus accessory proteins function. For example, VPU interferes with ApoBec, and then VIF can also interact directly with tetherin or BST2. So those accessory proteins expressed by lentiviruses, whether it's HIV, FIV, SIV, they all have a very defined anti-host function. I don't think endogenous retroviruses have that. And remember, they're a lot more stable, right? HIV and SIV they're mutating all the time. So it's a bit different situation. There are other proteins. I just don't know what they do. Yeah. Yes.
What's the population of these viruses? For example, SARS-CoV-2 in the brain. So is it like every square centimeter?
Yeah.
How many viruses there?
So actually, I had a slide. So we can get thousands of copies of RNA per gram of tissue, not millions, but thousands, upwards of 10,000. But the other thing I want to emphasize, it's not replication competent. Like I don't want you to think that it's actively replicating in the brain. The virus is there. And I think that's enough to induce an immune response in the brain. Just like sticking poly-IC in the brain is going to induce an immune response. But I don't think it's replication competent to any extent. I just don't think astrocytes have the right machinery to enable it to replicate the way other leukocytes and lung cells do.
So if you were to find, this is the last question, the status because cycle in astrocyte protein, 3 proteins, loading blood, this is on article, and is it always associated with the virus itself, or you get enough 3 proteins?
I can't say. Yeah, I don't know. I mean, I don't know. Francois may know more about that than I do. I really can't speak to that, Chris. Sorry. Yeah, it's an excellent question. Yeah. My sense is my sense, Chris, is a lot less protein than there is genome. Yeah. Oh, thanks, everybody. Thanks for the questions. And just remember who's going to win tonight, right?